Hi class, Dr. Searcy here with the lecture for Psych B1B Chapter 5. And as you can see, the topic of the chapter is vision. Just want to begin by noting that vision is one of the five senses that we enjoy. And in thinking about that, question that we want to answer is, well, where does this sense begin for us? When I experience vision, where does that first happen for me? In this, we're talking about a process called sense coding. And an answer to the question that we just asked is we could say that the sense of vision begins at the neuron receptors, right? And so let's just remember, we're talking about neurons throughout this class. Neurons are these microscopic organisms that communicate with each other. Specifically, we're talking about the dendrites on neurons. On those dendrites, we're seeing that we've got receptors. And remember, those receptors are there to pick up information or neurotransmitters from sender neurons, right? So if I experience vision, it's starting by some receptor on a dendrite receiving some stimulation or some information from another neuron. Okay, so another thing that we want to think about with that is this idea of the law of specific nerve energies. This is an idea that's been around since about 1838, and it basically states that whatever excites a nerve establishes a specific energy, right? And this is kind of the idea of what's going on behind the five senses. In the case of vision, there is a specific energy that we are detecting, right? So we have these receptors that are specifically there to give us information about vision. And the way that they do it is that they are sensitive to light. And so light is the kind of energy that these receptors detect. For the other senses, it might be different kinds of energy. For instance, like with hearing, it might be like sound waves or air moving through the air that's going to get detected, and there are certain receptors that are built for that. Okay, so with that, we also want to note that we're talking about this process that consists of three different steps: sensation, transduction, and perception. Where we can say that this first step, sensation, is where these receptors that we're talking about detect some light in our environment, right? So say you're sitting at a traffic signal, you are waiting for some signal to be able to go, right? You're waiting for the green light. Once you see the green light, you're going to be able to go. The idea is that the green light is what is making contact with your receptors when you experience sensation. Now, the idea is that light is a type of energy that your brain cannot interpret, does not understand. So your brain is going to have to translate, let's put that right here, is going to have to translate the meaning of the light or translate it into like a language that the brain can understand. That's going to happen by the brain transforming the light into a type of electrical chemical pattern, right? So remember, we were talking about how the brain works. We said that it works partly due to electricity and partly due to chemical stimulation. So because the brain works according to those two types of things, everything that stimulates the brain gets translated gets turned into that type of a pattern. So then once the brain sees that, then perception can occur, where perception is you understanding the stimulation, you understanding or having a sense of what the light is that you are perceiving. Okay, so with all of that then, the next thing I want to talk about is Well, how does this process occur inside the eyeball to begin with, right? And how does it then connect to the brain? So the thing that we want to do at this point is just kind of take a look at this diagram of an eyeball that I've got here. And I just want to describe the process of what all parts are in the eyeball that are going to be activated when vision occurs. So let's begin at the start, right? The start is where light enters the eyeball. When it enters the eyeball, it's going to come in through this outermost port called the iris, which is the colored part of the eyeball that we see, right? So like the brown or the blue or the green part that you see, that you say that is your eye color. By the way, this is something that is a product of evolution, right? Just like we talked about, I think it was in the last chapter, how different parts have evolved to adapt to the environment. What's evolved here is eye color itself, right? So think about when our species first began in Africa. It was really sunny there, and our eyeballs had a brown color to them. That brown was there to do a good job of collecting the abundant amount of sunlight that was available. Then we had other ancestors that moved into different regions of the world, like into the north. And the people that lived there developed, right, their eye color, the genes that instructed the eye color mutated into colors like blue and green, right, lighter colors. They evolved into lighter colors because there was less light up in those areas, and so the eyeball had to catch more light, and it was able to catch more light with those lighter colored eyeballs. So kind of interesting story about how the color of your eye is something that's evolved. Okay, but we're saying that when light enters the eyeball, it's coming through the iris, but specifically it's going to enter through the pupil, right? Because the pupil is a hole in the center of the eyeball. As it passes through the pupil, it's then going to go through the cornea and the lens, where the cornea is this outermost layer of the eyeball, and the lens is another inner layer of the eyeball. One thing that's kind of interesting about the cornea and the lens is that, so the light is coming through both of them. The cornea does not adjust, right? It does not stretch or do anything like that. However, the lens does. The lens will stretch to help us get a better sense of what the light is coming through, get a better focus on it, right? So cornea does not stretch, lens does stretch. If we're saying that the lens does stretch, what stretches it? Well, it turns out there are these things called ciliary muscles here and here, and they control the lens. They are what will either pull it apart or contract it together. Okay, so light's coming in, it's passed through the iris, the pupil, the cornea, the lens. It's going to continue to travel through the eyeball, right, through all this tissue that you're seeing here. And the place that it's going is to the back of the eyeball. This area that you're seeing in yellow, all along the back side here. That area in yellow is referred to as the retina. And light is going there because the retina is lined with receptors, right? And it's those receptors that are first going to detect the light that's coming in. Now, as it turns out, when we talk about the receptors that are there are two specific types of receptors. There are the rods and there are the cones. Okay, so the retina is going all across the back of the eyeball. It has receptors. The receptors are rods and cones. The reason why there are the two different types of receptors is because they are there to pick up two different types of light, or to detect two different types of light. Rods will detect low light, right? So like night light or night vision. And cones will detect when there's a lot of light, and when there is a lot of light, they're going to use that light to help understand detail and color with regard to the image that is being illuminated. Okay, so from there then, we're saying that there's this most sensitive part of the retina called the fovea. And the reason I'm saying it's the most sensitive is because it's that part of the eyeball that we use to acquire acute or detailed vision, right? So if you're looking at something, you're really trying to focus on it, really trying to get a good sense of a clear idea of what it is you're looking at, you are aiming your fovea at that thing to get that good look. Okay, so now one thing that we want to note is that at the very back of the eyeball, in this little area here, there's like an opening in the back of the eyeball. And that opening is referred to as the blind spot. By opening, what I mean is I actually got these two things I reversed right here. What I mean is the blind spot is this area where there are no receptors along the retina, right? There's like this little gap right there. And the reason why there's this little gap there is because there's this thing called the optic nerve that is going to leave the eyeball. And that optic nerve is carrying information that is going to exit the eyeball and go to different parts of the brain. Okay, so as it turns out then, this little chunk right here is really important. There's a lot of things going on. For instance, in that little chunk right there are the receptors that are picking up the information, and then there are also these neurons that are exiting the eyeball with the information that the receptors have picked up. So that being the case, we do want to take a better look at this little area right there. And I've got this little cutaway image right here of that little chunk of tissue, right? So this is an enlarged version of the back of the eyeball. You're seeing that this is that blind spot where there are no receptors around, and the optic nerve is exiting out the back. So as we take this closer look at this layer of retina then, right, this little layer right here, we're seeing that layer of retina has different layers of neurons on it. We can say that the layer that's maybe like the bottom layer at the very back of the eyeball is the layer where the receptors are, right? Those rods, those cones that we've been talking about. They are back there. You got rods, cones, going all the way across the eyeball. They're there receiving light information, receiving information about the light. Once they receive that information, they then send that information to this next layer of neurons, referred to as bipolar cells. And their job is basically to communicate the information the receptors picked up to other neurons. Specifically, though, one main group of neurons that are going to get this information are these cells called ganglion cells. And the reason why these are important is because their axon is actually what exits the eye. It's that optic nerve. And so what's really kind of interesting here is the way that you want to envision the neurons that are back here, right, is first think about, okay, let me remember what neuron anatomy is. We got dendrites, we got cell bodies, we got an axon, and then we got terminal buttons at the end of the axon. The way they're situated here is you have got the cell body down here, the dendrites down there, and then the axon is this huge, long axon that is going to come and end up leaving the eyeball. Okay, so what's exiting, what's moving out of the eyeball, is the axon of those ganglion cells, right? So the cell body is right here. Well, let's say the cell body is right here. Here's your dendrites, and then you got your axons exiting out. Okay, so that's the anatomy of the neuron. Now that we have this understanding of how the eyeball produces vision, we want to discuss, well, what is vision? What is it that we're seeing when we experience vision? So now I'm going to move to the next page, and we're going to talk about color vision, right? Because that's mostly how we see. We see in terms of color. We really don't see in terms of black and white. So let's think about what we're doing when we do see in color. Our cones, right, that receptor that picks up the detailed information, is detecting light within a certain range. Right, so what we're saying is we're detecting light that is detectable to us. And it's detectable to us because it's occurring in a way that we are sensitive to it. So let's look at what light in general looks like. Right, here's a table that kind of expresses it. And the first thing that we want to know is that light, or the amount of light, gets expressed in terms of wavelength. Right? And so with this, you want to think about maybe like the waves on a beach. You're standing on the beach and you have waves coming towards you. With that, you want to think about different kinds of waves that exist. You can have waves that are coming towards you that are short in length. Or you can have waves coming towards you that are long in length. And so if that's the case, we're expressing here a spectrum of different wavelengths of light that we can experience, from short wavelengths to long wavelengths, right? And the way that we're kind of expressing short to long is in terms of these numbers of, I think that's nanometers of light. What we're seeing is that this certain range of light that we can see exists between 350 nanometers of light to 700 nanometers of light, right? That's our range of our field of vision. And within that range of our field of vision, we're capable of seeing colors, beginning with the shortest wavelengths of violet to green to yellow to red, where the red ones are the largest wavelengths. Now, another thing that we want to think about with regard to this range of vision that we have is that we want to think about this range as being actually kind of a narrow range of vision. It's a range that occupies that amount of space on this other table of light, right? So we can see light that exists from here to here. That's this field of different colors. But there are other types of light that exist as well that we can't see, right? Like ultraviolet light, X-rays, gamma rays, infrared rays, radar, FM, TV, AM, AC. What's interesting about this is that even though we can't see a bunch of these wavelengths, there are animals that can see them. For instance, there are some birds that can see ultraviolet light, and there are some bats that are capable of using radar. One thing that's kind of interesting to me about these other wavelengths, right, is these are like radio signals, FM, AM, TV. Maybe this is like what your antenna can pick up. So what's fascinating to me with this then is to realize, holy mackerel, if I were to go outside right now, these wavelengths of light would be existing out there, right? Because if someone's playing their FM radio, there's that light that's out there moving around. I can hear it, but I can't see it. And to me, it's just fascinating. I wonder what it would look like if I were able to see it. I also think it's kind of interesting with the gamma stuff, right? So out here, it's kind of interesting to think about what an X-ray, if you were able to see in terms of X-rays, what that would look like. Would it look like where you can see through people's bodies, like when you go to get an X-ray at the hospital? With regard to gamma rays, right? If you guys are any of those Marvel movie watchers, you know the way the Hulk turned into the Hulk. Well, they got exposed to some gamma rays. It's kind of interesting to think about what they have that makes it so that the Hulk became the Hulk. Anyway, it's interesting to think about this huge range of light, and then to consider how that's what we're capable of seeing just in that narrow band. Okay, but from there, let's now think about, now that we understand how light works, let's think about how there's these different theories of how we process light and how vision is then produced. A first theory that we can talk about is the trichromatic theory. Right, first I should probably just explain the name. Tri is basically referring to the idea that there's a 3 that's happening there, regular triple. And chromatic is something that's referring to color. So this theory is basically saying something about 3 colors. What's it saying? Basically it's saying that humans are capable of, or we have cones that are capable of picking up 3 different types of colors. More specifically, 3 specific types of wavelengths of color. Right, and so, and we are sensitive to these different wavelengths. By the way, what are these 3 colors or these 3 wavelengths that we're capable of noticing with the different cones? The 3 are red, green, and blue, right? So we have cones that are specific to detecting either red light, green light, or blue light. When we experience vision, what we're actually experiencing is these cones, right? There's different cones for the different colors, but them all talking together then produces this combination of colors that we can talk about. By the way, I went online to try to look for some picture to illustrate this idea, and the only thing that I could come up with was this picture that I thought was kind of interesting. That was made up of different varieties of the 3 main colors that we're talking about here, right? So there's a bunch of different kinds of reds in here, greens, and then different kinds of blues in there as well. But then the next theory that we talk about vision is a theory called the opponent process theory. This theory says that color is perceived as paired opposites, right? So you have a pair of colors that you see at the same time, but what you're actually seeing is them being contrasted to each other. What are these pairs that we can talk about? Well, a contrast of white versus black, red versus green, or yellow versus blue. The idea is that this theory is supposed to explain why we experience things like negative after images, right? So like when you look, say, like at a flame for a long time, and then after you take your eyes away from the flame, there's this after image that gets left behind. This theory seeks to express why the after image has occurred. And I think I've got it right. No, I don't have it right there. Okay. Basically what